Genetics and developmental disorders. Chromosomes are the double-stranded DNA located in the nucleus. They contain the individual's genetic information. Chromosomes have a diploid number of 46, or 23 pairs. That's the number of chromosomes in most cells. There are 44 autosomes, 22 pairs, and 2 sex chromosomes, or 1 pair. The haploid number for chromosomes is 23. That's the number of chromosomes that are in the gametes, the sperm and the ova. Genes are on specific locations on the chromosomes, and they provide information for the production of protein. Mitosis and meiosis. These are the functions of cell division and replication, and these are the times that mutations commonly occur. Mitotic cell division. This is the cell division that takes place for the somatic cells, which are also called autosomes. They result in 2 identical daughter cells, 46 chromosomes, and again, that's your diploid number. Meiotic cell division. This produces the gametes, the ova and the sperm. One cell divides in 2 stages and results in 4 daughter cells, each containing 23 chromosomes. That's the haploid number. What is a karyotype? It's a picture of the paired linked chromosomes. Mutations. Mutations are caused by multiple things, such as an error in DNA replication. This results in permanent DNA changes. When it happens on the somatic cells, or the autosomes, the mutations are not inheritable because they don't make it into the sperm or the ova. So mutation of the sex chromosomes is inheritable. Types of mutations include insertion, deletion, duplication, inversion, and translocation, and I'm going to refer you to figure 6.6 in your text. Trait inheritance. The gene or allele pairs determine the traits that parents pass to their children. There's the genotype and the phenotype, as we spoke of in an earlier class. Mendelian inheritance patterns. Mendel is the man who developed the concepts of inheritance patterns that we use today. And these inheritance patterns can either be homozygous or heterozygous. Homozygous. There are identical alleles for the trait. The 2 members of the pair are identical. They match. An example would be blood. The genotype may be AA, but the phenotype is A. Heterozygous. There are 2 different alleles for a trait. Use the same example, blood. The individual may have inherited an A from one parent and an O from the other, so that's a genotype AO. The phenotype is A. Allele expression. Think back to the Punnett square discussion we had in an earlier class. There's dominant and recessive. Dominant genes are more likely to be expressed, and recessive genes are less likely to be expressed. Genetic inheritance patterns. That can be autosomal dominant, autosomal recessive, sex-linked dominant, and sex-linked recessive. Autosomal dominant. In terms of diseases, a genetic mutation has occurred on one allele, and that's a somatic allele. If either parent contributed the mutated gene to their child, the disease will be expressed. An example here is Huntington's disease. Okay, here is the Punnett square. In this situation, the B is the normal allele. The mother contributed two normal alleles, whereas the father contributed the B and also the beta. The beta is the mutated allele, and it is dominant. So in this case, each child has a 50% chance of having the disease. Autosomal recessive. In terms of diseases, a genetic mutation has again occurred on one allele, a somatic allele. If either parent contributed the mutated gene, the child will be a carrier. If both parents contributed the mutated gene, the child will have the disease. An example is sickle cell disease. Okay, here's our Punnett square again for autosomal recessive. Both parents had the B, which is normal, and both parents have a beta, which is mutated and recessive. So there's a 25% chance that the child is going to end up to be a BB. That means not carrying the trait at all. There's a 50% chance that the child is going to end up being a B beta, which means the child's a carrier. And there's a 25% chance that the child is going to have two betas, and that means they have the disease. Sex-linked mutations. These are mutations located on the sex chromosome, the X or the Y. Most are linked with the X chromosome. An example is hemophilia. Okay, here is the Punnett square for hemophilia. In this case, you can see that female children have a 50% chance of being without the trait or the alleles at all, and a 50% chance of being a carrier. The male children have a 50% chance of being without the disease or the trait at all, and a 50% chance of having the disease. There's just not enough Y genetic material to keep the disease from being expressed. When you look at the karyotype, the X chromosome is quite large and the Y is quite small. The Y chromosome does not carry much material on it.